Good time of the day to everyone. Thank you, David, so much for inviting me and giving this opportunity to share my research. And thanks for everyone for showing up. It's an amazing audience. Uh, so this is the place where we work. You can see it, hopefully, in the in the background of my slide. Cold Spring Harbor Labs. So we're trying to see how we can merge various branches of quantitative sciences in order to uh, understand brain function. And I will describe today the effort of merging um, kind of theoretical neuroscience, AI, and developmental neuroscience. And uh, so this is the idea which I just borrowed from Sebastian Sung, is that it's a hypothesis that your experiences, your uh, predispositions, your genome, your memories are stored in the connections between neurons. So it is kind of important to understand how those connections form and how they evolve, how the architecture of the brain was formed in the course of evolution. And of course, there are a lot of neurons in the cortex, about 10 billion neurons. And on average, they make about 10,000 synapses. So it's a network with enormous complexity, even in the cortex. Uh, so you can try to do this back of the envelope estimation is what is the information capacity of this network. And so I learned this formula in statistical physics, that information is the log of number of combinations. Uh, and yeah, so you can just take binomial formula and compute n is the number of neurons, s is the number of synapses per neuron, it's 10,000, and the number of combinations just given by number of neurons to the power uh, of the total number of synapses. And then if you apply this formula, then you will obtain the enormous number of 400 terabytes. So our cortices can uh, fit an enormous amount of information. And I understand that like 400 terabytes is difficult to grasp. So I converted it to movies, mostly for American audience. So it's about 45 years of continuous high definition video. So we can essentially remember all of our experiences within 45 years. So, which then sets up the interesting paradox, is that in the course of neural development, cortex is created from the genome from scratch. And the genome, it has about 1 gigabyte of information. So it's about like 3 billion base pairs, which is about 1 hour of video. So how is it possible to create this network which can store 400 terabytes from just a one hour, from 1 gigabyte of information in the genome? So that's an interesting question. So on one hand, the good news is that whatever differentiates us from mice is probably contained in a short instructional video of about 1 hour long. So it may not be so difficult to understand. On the other hand, there is some form of paradox. So how is this created? This enormous structure. And of course, the paradox is somewhat contrived because the genome contains the instructions. So the information in genome is really the Kolmogorov complexity, right? So the instructional program can be very short to create a cortex. For example, I could say that let's create a random network, which will be like a very short instruction. But like obviously cortical networks are not random, and so it's interesting to understand what is the impact of this expansion of information from genome to cortex that is produced in the course of neural development. So this discrepancy between the amount of information in the genome and cortex has been discovered a long time ago by Roger Sperry, who I'm going to mention, and then it was rediscovered several times in the course of studies of neural development. Finally, it was discovered by us. And it was actually very productive in terms of like shaping the views on the neural development, especially early on. And it led Regis Perry to hypothesize that you have to have gradients to save the molecular labels to set up the brain connectivity. So in addition to this expansion, which I mentioned, when the genome is expanded into the cortex, which is this red arrow, there's also the blue arrow, which is that when the cortex is compressed back into the genome. And of course, here I don't mean Lamarckian evolution. I mean just this blue arrow is the natural selection, right? Where the most fit brain, the most fit cortical architecture will be passed along to the next generation. So it is like an implicit form of encoding. So if you think about this way, then cortex in the course of evolution has undergone the large number of cycles of expansion and contraction, expansion and contraction. And and we, in this study, we try to understand what is the effect of this going uh, through the so-called genomic bottleneck. So the fact that you have to put brain architecture through this tight information, narrow information channel, we call it genomic bottleneck. So what is the effect of that on the brain architecture? And like one simple idea is that it is a constraint, and constraints are sometimes good for you. The best example of that I know is dogs' brains versus wolves' brains, right? So wolves' brains, for the weight matched wolf and dog, wolves' brains are larger by 25%. So the harshness of the environment in which wolves have to survive is somehow contributing to their intelligence. So maybe this will somehow help us to understand the principles how brains evolved. And so the idea here is that if you have to take this enormous network and compress it through the bottleneck into the genome for the next generation to inherit, you'll be probably very careful about selecting what are you going to take with you. So the idea is that putting the cortical architecture through genomic bottleneck has distilled the most important parts of the architecture and helped to create this powerful computing device that we have on our shoulders. So this is the genomic bottleneck hypothesis that postulates that genomic bottleneck that I just described led to the distillation of important information about statistics of the outside world and to the emergence of general intelligence. So there are many examples of how the genome can impact our brain function in the course of lifetime. For example, the learning rules, which are also encoded in the genome, could also shape the way how we process information. But like the immediate goal, which I'm going to describe in this study, is just neural development, production of the brain and the functioning of the brain right after it's born. And there are many ways, there are many interesting behaviors that animals display immediately after they're born. So it is kind of a complex problem. So our brains are quite capable right after birth. And I give here some examples. And of course, like many of us can relate to the concept of baby geniuses. But my favorite example is probably newborn turtles. You know, the turtles that are born, the eggs are lay in the sand, they're deposited in the sand, and then when they hatch, they wait for the night. They hatch. They wait for the nightfall, because that's when the predators are less likely to eat them. Then they come out of the sun, and then they run to the sound of the break of the shore break, and they obviously do it. They've never seen this world before, right? So all this behavior is encoded in the genome, and then the walking that this displayed in this wonderful video, it's just a newborn deer can do something that robots cannot do at this point. Obviously, spiders making webs, flying. And another very interesting question is the Chomsky's hypothesis about innateness of language. the idea of which that many aspects of language are encoded in the genome, and somehow that simplifies that gives us ability to learn languages from so many examples. So I'm just saying that innate behaviors are the impact of the genome on the innate behaviors, which are obviously encoded in the genome, is very interesting, and they are quite complex. So I'm going to address this in this talk. Of course, the question is, how is it possible? How can genes instruct connections? And then we have been working on this problem, like mathematical formulation of this problem, like since like in the early 2000s. And so this kind of idea, so we were trying to embody this principle that was postulated by Roger Sperry, whom I already mentioned. The idea of which is that cells express certain molecular labels, and then they roughly sniff each other, and they figure out whether they should connect or not, and they express multiple molecular labels. I'll just read the quote. The cells and fibers of the brain must carry some kind of individual identification tags, presumably cytochemical in nature, by which they are distinguished one from another almost in many regions to the level of a single neuron. And right, so this is some form of neural olfaction or chemoaffinity hypothesis. Is something that we've pursued for quite a while and tried to embody it in mathematical formulation. So, so this is like one example, which is probably the most studied. I'll just entertain you with some example of how chemoaffinity works. So this is cross section through the mouse retina. So this is the crystal we can see here, and sorry, so the mouse eye, and this is the retina in the back of the eye, and retinal ganglion cells is this layer, which are the output neurons of the eye. There are about 1 million of them. And what you can see here with this pink color is the expression level of a protein, which is called FA5. And this protein is actually important for setting up the topographic projection from the eye to the rest of the brain. And as you can see, the protein forms a gradient from the temporal part of the retina close to your nose, from the nasal part of the retina to the temporal part of the retina, and in such a way that this horizontal coordinate in the retina is encoded in the level of this protein. So it's like literally the X coordinate in the eye. So the model that we created explains the connection from the retina to superior colliculus, which is the region in the brain, in the vertebrate brain, which is responsible for eye movements, for one thing. So the axons from the eye, like of the neurons in the eye, they come out and they join the optic nerve, and then they reach superior colliculus, and they distribute themselves in a topographic manner. So there is actually an image of the retina, sort of like a TV screen in the superior colliculus. So there is a representation of X and Y in the superior colliculus of the retina. And so this is what is called the topographic map. And it's set up using those molecules. And so one of the ways how we approach this is by formulating this energy function, what the connection also is dependent on is on the expression of this other protein, which is called FRNA, in the target, which also has this gradient expression. And as you can see, the axons that have high level of AFA, they end up at the low level of FRNA, and vice versa. So the axons that are light in FA, they end up in high levels of FRNA. And so the idea is that there is a chemorepulsion. So the axons with high level of FA, they really hate high levels FNA. So there's a chemorepulsion in this receptor FA and ligand FNA pair. And the reason why not all axons don't collapse in this part of the target with a low level of FNA is because There is also competition between axons. So roughly speaking, they occupy a fixed area. So that forces them to distribute themselves over the target. So these axons that are more indifferent and they have low level of FA, they will end up in the most precarious place of high level of FNA. So to formulate this idea of chemorepulsion, we defined an energy, and like to map it on physics, I just used the charges and the electrostatic potentials here. So the charge is the level of this protein, Fa, and the electrostatic potential is phi is the level of the ligand for this protein, FNA, in the target. And the energy is just a product of, of them. And so paradoxically, repulsion actually comes with a plus sign. So if you minimize this energy, you will find that they will distribute themselves very nicely topographically along the XY axis. Well, of course, you can ask a question of what happens with the vertical axis. But the vertical axis, since the topographic map is two-dimensional, is controlled by just other two types of proteins, which are called FB and FRIN-B. And the logic is very similar, except where there is a schema attraction between FB and FRIN-B. So the axons that have high level of FB, they want to end up in high level of FRIN-B. So you can also set it up as an interaction of charges. Now there will be B charges. I mean, it's a colored charge, right? So it's a different type of charge with the letter B. And it's the sum over all axons of the charge carried by the axon by the level of ligand or electrostatic potential at the place where this axon terminates. And it comes with a minus sign because, again, paradoxically, the attraction has to come with a minus sign. Because if you minimize the energy, you want to maximize that product. So, the overall idea is that this simple Hamiltonian or the simple energy functional can help you set up the connectivity in this particular projection. We published a review about it. I mean, we published a lot of papers on this topic, which I reviewed there. And so we also think about how to generalize this to like more labels. I'm going to just go through some formulas here to describe our thinking. So this is the Hamiltonian which I just described to you. So this is the charges carried by axons times the electrostatic potentials, which are ligands at the place where the axons terminate. There are two types of charges. You can also collapse this formula if you introduce the interaction matrix between different labels. Like, for example, in this case, you see that the matrix is diagonal. So the charges type A interact only with charges type A, and charges type B, they interact only with charges type B. So I put this diagonal matrix there, M alpha beta, which is affinity matrix between different proteins. It doesn't have to be the case that it's diagonal, right? So they can be all diagonal interactions as well. So also you can introduce the weight matrix there. So the position where the axon terminates, where the electrostatic potential is determined, can be just replaced by weight matrix, which will allow me possibility for axon to terminate in many places, right? So this would be describing where the axon is making synapses. This is like more complex form of the model where you describe not necessarily one-to-one connectivity, but maybe more distributed connectivity. And you describe it by weight matrix. And then I'm rushing through this just because this slide is a form of advertisement for our theory. So if you are curious about it, you can read more about it in the review. So and then finally, you can also view this dependence on the weight matrix between the source and the target as the first term in the Taylor expansion of this energy. So you can include also the second term, which is proportional to the square of the weights. And the second term turns out to be responsible for Hebbian learning rules. So you can derive it from the Hebbian learning rules, in which case this matrix CIN becomes the correlations between activity of neurons, which are induced by retinal waves. So overall, to summarize, so how do genes instruct connections? So the idea is that genes create molecules. They make neurons express certain molecules, which are molecular labels by which they can recognize each other, and also the logic of the interactions, which could be described by that matrix gamma, alpha, beta, which I showed you before. And then that creates our cortices after in the course of neural development. So now we enter the age of AI. So I would like to now describe how we are going to approach this as an AI algorithm, this brain evolution problem and neural development. So now the idea is inspired by those molecular labels that I described before. So each neuron. So suppose that you want to create this phenotypic network, which is your cortex. This is the final product of the neural development. And there's an entry in this matrix with coordinates I and j. So this is a connection. This is a synapse between neuron I and j. So the neurons have to make a decision about whether to make that connections and how strong it is, right? So to create this connection, the synapse, we are going to have another network, which is called GNet. It's a genomic network, which is presumably encoded in the genome. The input to that network will be the arrays of molecular labels that similar to the ones which I described to you. Like, for example, this would be FA, this would be FB, in one neuron and another neuron, and then or this would be Ephrin A and Ephrin B in another neuron for that particular example that I described. And so you feed those, but they can be thousands of labels, right? So there are a lot of genes that are responsible for neural development. So they're fed into this GNet, which is hopefully much smaller than the PNet. So therefore the compression. And then this GNet will produce this strength of the synapse between two neurons. So this is like very machine learning way of thinking about it. But then what we're going to do is we're going to train PNet to do its stuff, whatever it is useful for. We give some inputs and we'll require some outputs. And we will backpropagate the loss from the PNet into the GNet. And that's therefore, by optimizing function of PNet, we will also optimize the GNet, which is much smaller. And so this backpropagation algorithm, it minimizes the loss, maximizes fitness, and it's a very non-biological form of neural evolution. So this is how we actually mimicked the evolution of GNets. But hopefully, if we find the right minimum, doesn't matter how we found it, whether we found it by our selection, inheritance and selection, or some Monte Carlo algorithm, or we found it using backpropagation. What I want to emphasize here is that in our approach, we don't store the molecular labels, we just store the GNet. And that allows us, because molecular labels are just generated from predefined set, so we don't have to store them. And instead, we ask GNet to wrap itself around to cater to those particular labels that we generate and to learn how to produce the most efficient PNet. So I'll give you one example of MNIST. Obviously, the first problem that everybody tries to solve is this one. Characters written by American high school students and just how to recognize them. So this is how we set it up. So we just use a two-layer MNIST network, which takes inputs in terms of like those written digits. The first layer, there's a hidden layer of 800 units, and then the output would be like the one-hot encoding of the labels. So there are two weight matrices. We're going to have a GNet. So this is our PNet. This is our cortex, right? Toy model of the cortex. And we are going to encode it in the GNets with quite a big compression, actually. We're going to squeeze it about 300-fold. And the GNet, as described here, is just like two fully connected layers. So there are two GNets for each layer of the network. to encode the labels. So these are the labels. There is the gray bars are representations of the labels that we feed into the network that represent individual neurons. So to represent the labels in the input layer, we'll just use like binary labels, but these are interesting binary labels. They're called gray codes. The idea is that these are binary numbers which are shuffled in such a way that the difference is one Hamming distance for the two nearest neighbors. So if you look at this, like for example, this one is different from this one by this bit and so on, right? So you can actually generate it in 2D or 3D or as many D as you want, but this is how we encode space through binary labels. Again, it's a toy problem. So there is no sense actually to have any kind of space in the hidden layer, so we just encode it by binary labels for appropriate number to encode 800 neurons. And so and then 100 encoding for the output layer. So and these are the results. And I think like it's important here because we have two times time axis here. One time axis is evolutionary time axis, and another one is the lifetime axis, so which is time after the network is born. What I'm showing here is the time after birth. So the blue curve is the conventional MNIST network, the same one as I described before, which is trained from scratch. It starts from 10% performance because it's just a random performance with 10 digits. And then it slowly rises to about 98% performance. But this is our network, which was born from the GNet with 300-fold compression, the red line. So the red line, as you can see, immediately after birth, it was already performing. It was able to recognize digits at about 95% rate. So this gives us hope that for larger networks, we can also, more complex problems, we can also distill the information and they will be born quite capable, from our GNets. So what is going on here? So the idea is that we have this is the space of the set of high performance networks, the red one, and the blue ones are the very compressible networks. So our algorithm tries to find the networks which are both high performance and compressible, and this is actually a small sliver, a very small subset of each of them. So for example, if you take high performance network and compress it, it's very unlikely that you will, we did this experiment, you will not be able to replicate that result, which I showed you of 95% performance with compression. So it's important to actually use backpropagation and iterate and co-train G and PNets together so that we find this part whereby we actually distill the important information. So I will show you, we apply our method to many other networks. You can read it in the paper, which we just published last year, including ImageNet, like some simple ResNet networks. I'm going to describe to you like a very entertaining example of reinforcement learning here, where we have this organism called half cheetah that exists in two-dimensional world. Well, it's constrained to move only it cannot topple over, right? It can only move in 3 dimensions. So in 2 dimensions, it receives. There are 18 variables which describe. There are 6 hinges that describe by 18 variables, and there are 6 control parameters which are torques in those hinges. And the goal is to make this cheetah run. So it's controlled by a simple 3-layer fully connected network. Which generates the policy, right? So they takes in 18 parameters and generate 6 torques. And we train it by reinforcement learning. The idea is to maximize the speed of running forward. And this is the example of cheetah, which is trained by conventional reinforcement learning, which is optimization the speed forward. So the example is on the right. As you can see, with no compression, it runs pretty well, but it kind of runs weird. And this is called reward hacking, where the network, when it's unconstrained, it will just find some kind of weird solution. In this case, it just slides on the chin, but it runs with pretty good speed. So this is the half cheetah, which is on the right. You can see the adult half cheetah, which is trained very well, which is initialized by the GNet with about 5-fold compression. But on the left, you can see the half cheetah, which was just born. It was just initialized. And it actually runs almost in distribution. It runs a little bit slower than the fully trained cheetah, just because you can see that it runs off the table a little bit slower. But it's almost performing at the level of adult performance. Uh, so this gives you an example that some more complex behaviors maybe can be learned with kind of can be encoded with substantial compression, in this case, 5-fold. But what happens if we put those cheetahs in the novel environment, such as the increased gravity environment? Suppose that they're born in the planet which has higher gravity than that planet that we trained it on. So this is the comparison of the trained cheetah on the left, trained cheetah without compression, and on the right is our GNet compressed cheetah. And because in most cases, the conventional reinforcement learning learns this reward hacked solution where it does something weird, it has difficulty walking, the cheetah on the left. However, the cheetah on the right, as you can see, is although it's kind of clumsy, I call it Jupiter shuffle, but it's still capable of propelling itself forward. So the idea is that GNets can learn some behaviors because of compression that can generalize across environments and can generalize to different tasks. I would like to switch gears here and to talk about this interesting problem. And it's like a very recent development is how to learn distribution of labels. I described you the distribution of molecular labels as binary, gray codes, or Manhattan coding, but it could be also just the real numbers. But well, how can we discover them? And the idea, of course, is to have another network. In this case, it's f of x that will encode the molecular labels. So the idea is that you feed some random numbers distributed in a queue to this F network, and that this network that learns the labels can hopefully shape them in the right way that is suitable for particular tasks. So we are going to discover the labels. So the only parameter that we have here is the dimensionality of this queue, of this random number. In this case, it would be 3, right? So it would be 3-dimensional random numbers. And again, we backpropagate the loss of the PNet, which is our cortex, to the genome and then to the label encoding network. So again, I'm going to go back to the example of MNIST network. We have the space encoding gray codes, but instead of just using binary labels for the hidden layer, we're going to use n-dimensional random number. It used to be just binary number, but now it will be random. And we're going to vary the number of dimensions. And this is time, but in this case, it's evolutionary time. So this is training the GNet. So it's roughly speaking, describes the generation of GNet. So there are different performances. Each of them describes now performance at the network initialization. And what you can notice here is that one-dimensional, if the random numbers that encode the molecular labels are random one-dimensional numbers, then it performs, but not so well. So the final performance is less than 80%. Two-dimensional random numbers also are not doing so well. But like after the 3 dimensions, you essentially saturate the performance. There's no change in performance after the space of labels goes from 3 to 4 dimensions, which means that essentially the space of cell types in this hidden layer has to be three-dimensional, because it makes no sense to add an extra dimension. If the network was evolving, it would take additional time to encode this extra dimension. So this addresses the issue of the space of cell types in the hidden layer in particular. And so we find for some reason that for MNIST network, it's 3D. I mean, it's an open theoretical question. I mean, so far, it's just a numerical result. But interestingly, it correlates with this finding, which was published by the late great Chuck Stevens around like 2001, I think, that if you look at the scaling of retina or LGN of cortex versus LGN or retina or LGN, they scale with the number of cells. So they're roughly two-dimensional. So they just expand. As you add more cells in the retina, LGN will just blow up. And but the cortex expands faster, which suggests that cortex, so Chuck suggested that cortex, the space of cell types in the cortex may turn out to be three-dimensional. Because the scaling law is 3/2. So which means that cortex, if retina or LGN is two-dimensional, then cortex, it encodes one extra dimension. So it seems that the result that I presented before, the numerical result, correlates with this idea, but I don't really know at this point how to wrap my head around this problem, like theoretically, and discover this solution. Anyway, so this also offers the opportunity to simulate something which I call neurogenesis, where you sample, you add more and more neurons to the network from this box, from the three-dimensional box, while you train the GNet, while the network evolves, and hopefully you will be able to discover, as you add more neurons, you will be able to discover new features in your network, because network will become more complex. So this is just an example of neurogenesis in the MNIST network, where every 100 GNet generations, I will just add extra 100 neurons to the middle layer, to the hidden layer. And the remarkable thing that eventually it will learn the correct distribution of labels. So even though I just like add random neurons to it, it doesn't really hurt the performance. If anything, the performance is increasing. So I think it's a very interesting problem. And I'm hoping that at this point, at some point, I will expand it to more complex networks. So I probably have like around another 5 minutes. So I will talk about some fun stuff about human evolution. So I read this book, which is called Culture and the Course of Human Evolution, from the suggestion of Tatiana Sharpie. And, I mean, it's a very interesting book, which just analyzes how our cognitive abilities have been changed in the last 150,000 years or so. And the idea there is that there is this niche type interaction between, so brain and culture, they co-evolve. So they form a niche. So there's interdependence between them. And brains, of course, they go through the bottleneck, as I described before. And like one of the examples of that interaction, of that niche, probably the most remarkable example is the expensive tissue hypothesis. I don't know if you've heard about it, but there's the idea that a lot of our intelligence is produced by the cooking food. So you know that our brains consume about 17 watts of energy, and about 4 of those watts, they come from cooking food. So the idea is that our organism is given about 70 watts of energy, and most of that energy actually goes for digesting food. So what if you can reduce energy from food digestion and add it to the brain? So when people started using fire in controlled fashion and cook food, they acquired those additional 4 watts of energy, which was transferred from digestion into the brain function. And that expanded our cortices by about 25%. So one can argue that Prometheus, in addition to giving us fire, he also gave us 25% of brain volume. So that was his real crime. So the question now is like I described to you the bottleneck in the brain evolution. Can the cultural phenomena form bottlenecks? And this is like one of the best examples of bottlenecks is physics. You know, all those remarkable phenomena that we see around us, uh, physics compiles them into short, mathematically concise descriptions. And so you can view physics as some form of autoencoder, where you look at the reality and you can actually explain the reality using simple equations, right? And the reason why this happens, besides the ability to generalize, is our ability to transfer the knowledge of physics to the next generation. So it takes maybe about like 10, 15 years to train a new physicist, as opposed to, or, you know, or chemist, because the same applies to other quantitative sciences. So, so the idea of calculus and physics are also bottlenecks, although they exist in the domain of culture. Um, another, so these two things, brain and culture, they can be argued to be self-replicating quantities. So the idea is that if you, for example, remove certain part of culture, such as if you remove English language completely, it will be completely wiped out. So that part of the brain will not be sufficient to replicate, to reconstitute English language, just because it is not really present in the brain. So, and vice versa, right? So if you wipe out certain species, it will be like very difficult using culture to reconstitute them. So these things are kind of self-replicating. So they require themselves to produce the next generation. So another example is tools, right? Tools are also self-replicating because to make more sophisticated tools, you need to have tools as well, right? You cannot use it like a stone axe to make a space shuttle. And so the tools, they evolved over time, and I'm putting here, this is, I think, A100 GPU. This is like one of the ultimate tools that we use right now. And I should say that tools also form a niche with the other two things, with brains, culture, and culture. So in the way that they co-evolve. But in this co-evolution, and this is my last point, brain is actually not necessary. So those two things can very happily self-replicate into the future. So um, on this happy note, I will offer you my conclusions. So I argued that the information available in the genome is not sufficient to set up cortical connectivity with single synapse precision. So, which is something we call a genomic bottleneck. And then I hopefully convinced you that genomic bottlenecks can force neural nets to distill architectures which generalize across environments and tasks. And they can ultimately lead to the emergence of general intelligence. So I would like to thank my collaborators, Tony, Sergey, and Divyansha, uh, for their very hard work and very stimulating and exciting discussions, and the support from Deep Valley Labs and Mothers Foundation. And thank you very much for your attention and for the kind invitation.